Population. A population is a group of members of the same species in a given geographical area who are potentially capable of mating and producing fertile offspring. Gene pool consists of all the alleles in all the individuals that make up a population. The process of meiosis, that is specifically crossing over, and fertilization shuffle alleles in the gene pool, giving us genetic diversity. The gene pool is a one that is in stasis. This is described by Hardy-Weinberg theorem. The frequencies of alleles in a population's gene pool remain consistent over the generations unless acted on by agents other than sexual recombination. Gene frequency. Proportion of different alleles of a gene in a random mating population is referred to as gene frequency. Estimation of gene frequency in a population consists of three important steps: sampling, classification, calculation of gene frequency. Sampling, classification, calculation of gene frequency. Genotype frequency. Genotype frequency at a specific locus can be calculated by counting number of individuals with particular genotype and divide this number by the total number of individuals in the population. Then genetic drift. The change in the frequency of an allele in a population due to random sampling of organisms. Common in small populations, not significant in large population. Some alleles some alleles become more common while others become less common over time or lost. When there is only one allele is left for a particular gene pool, that allele is said to be fixed. Genetic drift, random process, doesn't occur due to environmental challenges. Non-directional, non-adaptive evolution, important alleles may disappear, operate on neutral allele, loss of genetic variation. Once again, genetic drift, random process, doesn't occur due to environmental challenges. Non-directional, non-adaptive evolution, important allele may disappear, operate on neutral allele, loss of genetic variation. Next topic, types of evolution and evidences for evolution. Evolution or change over time is the process by which modern organisms have descended from ancient organisms. Evolution also explains how modern organisms have descended from ancient organisms. Evolution is the process by which organisms pass on traits from generation to generation and explains the origin of new species and diversity. In biology, evolution is the change in heritable characteristics of biological populations over successive generations. These characteristics are the expressions of genes which are passed on from one parent to offspring during reproduction. Genetic variation tends to exist within any given population as a result of genetic mutation and recombination. Evolution occurs when evolutionary processes such as natural selection, including sexual selection and genetic drift, act on this variation, resulting in certain characteristics becoming more or less common within a population over successive generation. It is this process of evolution that has given rise to biodiversity at every level of biological organization. Evolution, descent with modification. Evolution means change over time. Evolution can also be defined more narrowly as a change in the genetic composition of a population from generation to generation. Evolutionary descent with modification, formation of newer types of organisms from the pre-existing ones through modification. It is an orderly change from one condition to another. Types: progressive, retrogressive, micro, macro, mega, parallel, convergent, divergent, and coevolution. First, microevolution. Microevolution is a change that occurs in time in population due to changes in allelic frequencies. This happens due to four reasons: genetic drift, mutation, gene flow, natural and artificial selection. Examples of microevolution: the size of the sparrow. Sparrow populations in the north are larger bodied than sparrow populations in the south. This divergence in populations is probably at least partly a result of natural selection. Larger bodied birds can often survive lower temperatures than smaller bodied birds can. Colder weather in the north may select for larger bodied birds. Then macroevolution. Macroevolution brings bigger, large scale and visible changes above species level. It is interspecific in nature. The new changes are often as big as development of a new structure or organ. Macroevolution usually means the evolution of large scale structures and traits that go significantly beyond the intraspecific variation found in microevolution, including speciation. In other words, macroevolution is the evolution of taxa above the species level, genera, family, orders, etc. And macroevolution is often thought to require the evolution of completely new structures such as entirely new organs. However, fundamentally novel structures are not necessarily for dramatic evolutionary change. For instance, the evolution of a mammal diversity in the past 100 years has not required any major innovation. All of this diversity can be explained by the modification of existing organs. And examples of macroevolution: Homo sapiens. Human macroevolution from our ape ancestors is indicated by fossil records. So, examples of the fossils that report the theory of Homo sapiens macroevolution include Homo habilis, Homo rudolfensis, and Homo erectus. Australopithecus, Homo habilis, Homo erectus, Homo neanderthalensis, and Homo sapiens sapiens, and Homo rudolfensis. Macroevolution: large scale changes in gene frequencies. Microevolution: small scale changes in gene frequencies. Macroevolution: longer time period. Microevolution: few generations. Macroevolution: at or above level of species. Extended microevolution: not directly observed. Fossil evidence: more controversial. Example: reptiles to birds. Then microevolution: small scale changes in gene frequencies. Few generations within species or population. Small evolutionary changes observable. Environmental evidence: less controversial. Example: bacterial resistance to antibiotics. Then convergent evolution. In evolutionary biology, convergent evolution is the process whereby organisms not closely related independently evolve similar traits as a result of having to adapt to similar environments or ecological niches. When animals come from different ancestral lineages caused by environmental changes, also called convergence. An example: shark and dolphin bodies, vertebrate and cephalopod eyes, the relationship between bat and insect wings. Examples: shark and dolphin bodies, vertebrate and cephalopod eyes, the relationship between bat and insect wings. Convergent evolution: shark, ichthyosaur, dinosaur. Dolphin. Not dinosaur, dolphin. Then analogous structures. Convergent evolution creates analogous structures that have similar form or function but were not present in the last common ancestors of those groups. Organisms have same function but not same the structure. Analogous structures arise from convergent evolution. Convergent evolution of echolocation. Both bats and whales use echolocation to find food. Bats hunt for food at night and use echolocation to see. Whales have limited access to sunlight in the ocean and use echolocation to find their way around. Convergent evolution of echolocation. And divergent evolution. Divergent evolution is defined as that what occurs when two groups of the same species evolve different traits within those groups in order to accommodate for differing environmental and social pressures. So species gradually become different from its ancestors. And evolution results in multiple different species leading to speciation. Homologous structures developed, also called adaptive radiation, caused by environmental changes and migration. Similar, all have vertebrates, all have mouths, all have eyes, developed from sexual reproduction. Most eat and drink, react to environmental ability to move. Different colors, some have fur and hair, some live on land, some can swim. How they move? Environmentally, they live in interaction with other species. Then examples of divergent evolution: Darwin's finches. Finches on the Galapagos Island evolved into many different species to eat different foods: polar bear and brown bear, red fox and kit fox. Then homologous structures. In divergent evolution, homologous structures are structures indicating a species is diverged from its ancestor. The structures need not have the same function as that of those found in the species ancestors. Same structure, different function. Homologous structures arise via divergent evolution. Same structure, different function. Example: the forelimbs of humans and bats, homologous structures. Man, cheetah, whales, bat. And parallel evolution. Some species evolve without converging on similar traits or diverging into different traits. The species undergo change, but they maintain a constant level of similarity to each other. This process is known as parallel evolution. Parallel evolution occurs when two species evolve independently of each other, maintaining the same level of similarity. Parallel evolution usually occurs between unrelated species that do not occupy the same or similar niches in given habitat. Marsupials and placental mammals most recently shared common ancestor 125 to 150 million years ago, yet this similar features appeared in each of these distant relatives. The good example is a parallel evolution is in elephants and extinct mammoths. Both animals are closely related, but their similarities evolved independently. And coevolution. Coevolution is the process by which two species evolve in response to changes in each other. Species that live in a close relationship with each other, such as a predator-prey or symbiotic relationship, often evolve adaptations to each other in a process called coevolution. Then types of coevolution: parasite-host coevolution, mutualistic beneficial coevolution, predatory-prey coevolution, etc. Then mutualistic or beneficial coevolution. Yucca moth, Tegeticula eucasella, a female yucca moth pushing pollen into the stigma tube of the yucca flower while visiting the flower to deposit her eggs. Predatory prey evolution. One common example is the relation between some species of birds and butterflies. And parasite host coevolution. An organism lives on other organism, harming it and possibly causing death. The parasite live on it in the body of the host. Maize plant host and oospose is a parasite, core maize disease. And evidences: anatomical, morphological, embryological, paleontological, physiological, biochemical, cytological, molecular, then biogeographical, taxonomic, and genetical evidences. Then anatomical or morphological evidences. Anatomical or morphological evidences in the sense, study shows that some features are basically similar: anatomy and morphology, body organization, gradual modifications, etc. It includes homologous organs, analogous organs, connecting links, vestigial organs, and atavism. And homologous organs in detail: similar embryonic origin and structure but differ in external form and function. It shows the adaptive radiation from a common ancestor to the give evidence to divergent evolution. Like vertebrate forelimbs, insect legs, insect mouth parts. Forelimbs, same general function of locomotion but have different shapes and bring about different types of locomotion. Legs of insects: coxa, trochanter, femur, tibia, and tarsus. These are the regions. Then evidence from comparative anatomy: vertebrate limbs. The basic pattern of the vertebrate limb is represented by the human arm skeleton shown below. Ball and socket joint, hinge joint, single bone, two bones, group of small bones, five joint digits, etc. can be seen. Then comparative anatomy: homologous structures, anatomically similar structures inherited from a common ancestor. Comparative anatomy: human, whale, dog, bird. Then types of homology. Phylogenetic: this is the homology between different species of animals. Sexual homology between the sexes of two samples or species. Serial homology: in the serial segmentation of the body of different groups or species. Arthropods. Analogous organs have similar structure and superficial appear similar and are different in fundamental structure and embryonic origin. For example, wings of insect, birds, bats, etc. Then vestigial organs: degenerate, functionless, underdeveloped organs that are similar and smaller than the corresponding organs in ancestral forms. They are not useful and necessary for their possessive thought, though might have been well developed and functional in the distant ancestral forms. The presence of vestigial organs explains that during the course of evolution, animals change their habitats and modes of life. Vestigial organs: appendix in humans and internal hind limbs of whales, and the internal legs of some species of snakes like python and boa constrictor. Large intestine, small intestine, appendix. Then vestigial structures: the skeleton of a whale reveals the presence of pelvic bones. Pelvic bones, subbones resemble those of the mammals but are only weakly developed in the whale and have no apparent function. And embryology reveals a unity of plant. Embryology of many vertebrates exhibit many common features in development. The early embryos of life. Different vertebrates appears have two very much similar in the external form and structure. Subsequent replacement of notochord by feces. Similar development of limbs from limb buds in tetrapods. Next, biogenetic law. The theory of recapitulation, also called biogenetic law or embryological parallelism, often expressed using Ernest Haeckel's phrase ontogeny recapitulates phylogeny, a historical hypothesis that is the development of embryo of an animal from the fertilization to the gestation or hatching. Ontogeny goes through stages resembling or representing successive adult stages in the evolution of animals' remote ancestors, or phylogeny. It was formulated in the 1820s by Tynsters based on the work of John Friedrich Meckel, after whom it is known as Meckel-Serres law. Then fossils, paleontology, Da Vinci, George Cuvier. Then organic remains like bones, hairs, nail, claws, shells, impressions of organisms found in sedimentary rocks are called fossils, or they are the remains of extinct organisms buried and preserved by natural processes. Paleontology and botany and paleozoology. Darwin first shows that fossils provide direct evidence for evolution. Methods of fossilization: intact preservation, petrifaction, molds and casts, impressions, mummies, and tracks and trails. Evidence for evolution: here could see a picture of Archaeopteryx fossil, half bird, half reptile. Claws, wing-like forelimbs, teeth, thin ribs, long feathers, bird-like characters. Bird-like characters, wing-like forelimbs, thin ribs, feathers. Reptile-like features, claws, teeth, and long tail. Inferences from fossil study. By studying the fossil's record, we can tell how long life has existed on Earth and how different plants and animals are related to each other. Often we can work out how and where they lived and use this information to find out about ancient environments. Fossil can tell us a lot about the past. Then fossil record. Fossils provide a record of species that lived long ago. Fossils show that ancient species share similarities with species that live on Earth. Fossils show that ancient species share similarities with species that now live on Earth. Then age of fossils, dating of sedimentary rocks, geochronology, dating system, clocks of rocks, carbon dating method, Libby. Then radioactive clock method, K-Argon method. Then living fossils, plants, animals which have become extinct except one or two representatives. Then types of radioactive dating. Potassium argon dating is used for rocks that are suspended millions of years old. Then the older rocks, the ratio of argon to 40K will be higher than in younger rocks. Carbon dating, two types, 14C to 12C. Radioactive 14C, non-radioactive 12C, most common. Then biogeographic evidence. Study of distribution of animals and plants in different parts of the earth. Alfred Russel Wallace, 6 geographical realms: Paleo-Arctic, Oriental, Ethiopian, Australian, Nearctic, Neotropical. Wallace line, Oriental, Australian. Then Weber line, further east than Wallace line, based on discontinuous distribution. Descendants of common ancestors inhabit different contents, mutated and slightly different but closely related species of the same genus. Then restricted distribution, the continent has particular fauna and flora. And physiological or biochemical evidences: metabolic processes, enzyme, hormones, blood, lymph, blood protein, blood groups, molecular hormones. And metabolic processes: digestion, biosynthesis, respiration, muscle contraction. Enzymes, alike in nature and action. Hormones, chemical nature and action. common ancestry can be seen in the complex metabolic molecules that many different organisms share. Organisms with closely related morphological features have the closely related molecular features. Classic example for biochemical evidence is the variation of cytochrome C in living cells preserved in all eukaryotes. Sequence of amino acids vary with a species at the same time. Its variants are due to evolutionary changes in use. And taxonomic evidence. Evolutionary relationships are reflected in taxonomy. Taxonomy is an artificial hierarchical system showing categorical relationship between species based on species defining characteristics. Each level within the taxonomic system denote a greater degree of relatedness to a particular organism if it's closer in the hierarchical scheme. Then organisms are usually grouped together based on their unique characteristics. The classification of an organism often provides useful information about the evolutionary history and with other organisms can relate to it. And phylogenetics is a fundamental tool for organizing our knowledge of the biological diversity we observe in our planet. A phylogenetic tree, also known as a phylogeny, is a diagram that depicts the lines of evolutionary descent of different species, organisms, or genes from a common ancestor. Evolutionary taxonomy, including evolutionary systematics or Darwinian classification, is a branch of biological classification that seeks to classify organisms using a combination of phylogenetic relationship or shared descent, then progenitor descent and relationship, or serial descent and a degree of evolutionary change. The phylogenetic principles of classification is an evolutionary principle in contrast to the phenetic principle. It classifies species according to how recently they share a common ancestor. The DNA evidence. Another important line of evidence for evolution comes from DNA analysis. Organisms which have much of their DNA in common must be closely related. They are split from a common ancestor comparatively recently in geological terms. For example, humans and chimpanzees have 99% of their DNA in common, while it's just a close relationship between them and a relative recent divergence from a common ancestor. Next is about Hardy-Weinberg law. Proposed independently by a British mathematician, G.H. Hardy, and a German physician, Wilhelm Wilberg, independently in 1908. So the law states that the alleles and genotype frequencies in a population remain constant generation after generation if there is no selection, mutation, migration, or random drift. In a finitely random mating population, the frequency of genes and genotypes remains constant generation after generation if there is no selection, mutation, generation, and random genetic drift. The 7 assumptions underlying Hardy-Weinberg equilibrium are as follows. Organisms are diploid. Only sexual reproduction occurs. Generations are non-overlapping. Mating is random. Population size is indefinitely large. Allele frequencies are equal in sexes. There is no migration, mutation, or selection. Allele frequency equilibrium, genotypic frequency equilibrium, neutrality of equilibrium. First, allele frequency equilibrium. Allelic frequency at an autosomal locus in a population will not change from generation to generation. Then genotypic frequency equilibrium. The genotype frequency of a population are determined by the allelic frequencies in a predictable way. Then basic requirements for equilibrium: random mating, large population. All genotypes have equal chance of survival and reproduction. Populations must be free from mutation. Population must be reproductively isolated. Random mating. The Hardy-Weinberg principle states the population will have the given genotypic frequencies called Hardy-Weinberg population after a single generation of random mating within the population. When the random mating assumption is violated, the population will not have Hardy-Weinberg proportions. A common cause of non-random mating is inbreeding, which causes increase in homozygosity for all genes. Non-random mating. Random mating is essential for Hardy-Weinberg equilibrium. When phenotypic genetic relatedness influences mate selection, it will lead to alteration in genotypic frequency but without alteration in allelic frequency. Then assortative mating based on phenotypic characteristics, but pairing is not random. And males of a particular kind breed only with the females of a particular kind. It can be positive or negative. Positive assortative mating phenotypes of mating partners are more similar. Positive assortative. The negative assortative mating between individuals with dissimilar phenotypes. For example, mating between individuals with matching height, that of mismatching height. Genetic relatedness, in and outbreeding. Inbreeding between genetically related individuals, outbreeding unrelated. Positive assortative mating, inbreeding, increased homozygosity. Negative assortative mating, outbreeding, increased heterozygosity. Then selection in general cause allele frequencies to change, often quite rapidly. While directional selection eventually leads to the loss of all alleles except the forced one, unless one allele is dominant, in which case recessive alleles can survive at low frequencies. Some forms of selection, such as balancing selection, lead to equilibrium without loss of alleles, while directional selection eventually leads to the loss of all alleles except the favored one, unless one allele is dominant, in which case recessive alleles can survive at low frequencies. Some forms of selection, such as balancing selection, lead to equilibrium without loss of alleles. Then mutation will have a very subtle effect on allele frequencies. Mutation rates are of the order 10 raised to minus 4 to 10 raised to minus 8, and the change in allele frequency will be at most the same order. Recurrent mutation will maintain alleles in the population even if there is a strong selection against them. Mutation will have a very subtle effect on allele frequencies. Mutation rates are of the order 10 raised to minus 4 to 10 raised to minus 8, and the change in allele frequency will be at most the same order. Recurrent mutation would maintain alleles in the population even if there are strong selection against them. Then small population size can cause a random change in allele frequencies. This is due to sampling effect and is called genetic drift. Sampling effects are most important when the allele is present in a small number of copies. And migration genetically links two or more populations together. In general, allele frequencies will become more homogeneous among the populations. Some models for migration inherently include non-random mating, Buchland effect, for example. For those models, the Hardy-Weinberg proportions will normally not be valid. Then in general, a gene contains two alleles, a dominant one and a recessive one. Random unit of gametes, consider the equation P plus Q is equal to 1 in case of alleles. Q is equal to frequency of allele A, dominant. Q is equal to frequency of the allele small e, recessive type. In case of genotypes, the Hardy-Weinberg equilibrium is represented by the equation P square plus 2PQ plus Q square is equal to 1. P square represent frequency of genotype AA, homozygous dominant. Q square represent frequency of genotype small a, homozygous recessive. Then frequency of genotype AA, heterozygous genotype. P plus Q is equal to 1, allelic perspective. P plus Q into P plus Q is equal to 1 square, genotypic perspective. So P plus Q, the whole square, is equal plus 2PQ plus Q square is equal to 1. P square is equal to PP, homozygous dominant. 2PQ, heterozygous. Q square, QQ, homozygous recessive. So remember this one, P square plus 2PQ plus Q square is equal to 1. Then results of random mating in a population, two alleles are given. What will be happening? P plus Q plus R whole square, P square plus Q square plus R square plus 2PQ plus 2PR plus 2QR. Then significance of Hardy-Weinberg law. Not only gene frequencies but also genotype frequencies of different alleles in a population remain constant. A population in Hardy-Weinberg equilibrium does not show evolution. In other words, for evolution to occur, the population should not follow Hardy-Weinberg equilibrium. All genotypes in a population reproduce equally and successfully. Then genetic drift, changes in allele frequencies due to chance, acts in a small population, reduce genetic variability. Genetic drift, change in allele frequency due to chance, acts in small population, reduces genetic variability. Example, bottleneck effect and founder effect. Bottleneck effect, it occurs when there is a sudden sharp decline in a population size, typically due to environmental factors, natural hazards such as earthquakes or tsunamis, epidemics, and can decrease the number of etc. Some genes which are there is not any distinction are extinguished from the population. Reduction of the total genetic diversity of the original gene pool. Surviving population is further from the original in its genetic makeup. Here you could see a population, the parent population, almost equal population of this yellow ball and the violet ball. But later, when the bottleneck effect is happening, a small amount of population is transferring into another bowl, that is the bottleneck. Drastic reduction in population because of some natural hazards or something else. Then surviving individuals are shown. Then next generation to a greater extent will be having more number of violet bubbles, because that is something which survived in the surviving individuals. Then founder effect, loss of genetic variation that occurs when a new population is established by a small number of individuals that are cleaved from a larger population. Founder effect, loss of genetic variation that occurs when a new population is established by a small number of individuals that are cleaved from a larger population. New population does not have the genetic diversity of the previous one. New population does not have the genetic diversity of the previous one. Some genetic traits are becoming more prevalent in the population. Once again, founder effect, loss of genetic variation that occurs when a new population is established by a small number of individuals that are cleaved from a larger population. New population does not have the genetic diversity of the previous one. Some genetic traits are becoming more prevalent in the population. Then leads to the presence of certain diseases, genetic diseases in the next generation. Founder effect, mother population, new population. Natural selection, next topic. The natural selection is a process by which organisms are better adapted to their environment, tend to survive and produce more offspring. The natural selection is a process that results in the survival and reproductive success of individuals or groups best adjusted to their environment. The process of natural selection relies on a few important factors like variation within a species is necessary. Individuals must vary in appearance or behavior. The variable traits must be inherited by offspring. Then examples of natural selection: Galapagos finches, beak size and shape adapted to a specific type of food. Their beaks evolved due to natural selection. Individuals with the right shaped beak to reach the food would survive. Galapagos finches, beak size and shape adapted for specific type of food. Their beaks evolved due to natural selection. Individuals with the right shaped beak to reach the food would survive. Then directional selection, disruptive selection and stabilizing selection. First, directional selection, the type of natural selection in which the phenotype of the species tend toward one extreme, directional, one direction. Just remember like that. Then occurs when natural selection favors one extreme of continuous variation. The favored extreme will become more common. Directional selection, one extreme trait is favored, then others are neglected. Then examples of directional selection: the frequency of the melanic form of the moth increased. Their darker coloration provided camouflage against the sooty tree. Had a higher survival rate in habitats affected by air pollution. Directional selection example: peppered moths. Allele frequency shifts to one direction. Shifts in one direction. Peppered moths example. Then disruptive selection. A type of natural selection that removes individuals from the center of a phenotypic distribution and causes the distribution to become bimodal. Bimodal in the sense both the extremes are removed, only the middle portion will be selected. Selection against the mean. So a type of natural selection that removes individuals from the center of a phenotypic distribution and causes the distribution to become bimodal. Occurs when natural selection favors both extremes of continuous variation. No, here both extremes are added. Then the two extreme variations will become more common, that is disruptive. Examples of disruptive selection: light colored oysters are more colored, allow organisms to match its background. Dark colored oysters blend into the shadows cast by the rocks. Intermediate colored oysters would be most heavily preyed upon the crabs. Then stabilizing selection removes individuals from both ends of the phenotypic distribution, maintaining the same distribution mean. Occurs when natural selection favors the intermediate states of continuous variation. The intermediate states become more common. Then example of stabilizing selection: very light colored or very dark colored oysters might be more frequently preyed upon by shore birds because they are more obvious than the oyster bar. The intermediate colored oysters become more common. Stabilizing selection, bidirectional selection, disruptive selection. Selection begins after selection. Then phenotype being selected for, phenotype being selected against. Stabilizing selection, middle one will be selected, mean portion. Then directional selection, one of the extremes are selected. Then disruptive selection, only extremes, both extremes are selected, not the middle one. Then the process through which males and females attempt to maximize their chance of reproductive success. There are two different types of sexual selection: intrasexual selection, intersexual selection. So intrasexual selection, mate competition. Members of one sex compete against each other to attract members of the opposite sex. Usually this is between two or more males. Intrasexual. Then intersexual, mate choice. Females choose between mates between sexes. And intersexual selection: in most mammalian species, the female invests heavily in the offspring, especially in species where females have limited reproductive opportunities. This female need to be picky and make the right selection of male. The wrong choice of a female is much more serious than for a male. Like this lead to intersexual selection. And sexual selection: the different genders in a species exert forces on each other that changes their appearance or traits. Natural selection favors traits that maximize the ability of an individual to compete, attract mates, the ability to produce offspring. And examples of sexual selection: the peacocks with the showy feathers are able to attract mates. Then peacocks can fold up their tails in danger or predator. Peahens are much duller and much duller and more camouflaged. They are the ones who guard the eggs and chicks. Survival of the next generation is very important. Then intrasexual selection. Intrasexual selection is a competition between members of the same sex, usually males, for access to mates. Between the members of the same sex, within, males fight with each other to chase others away. Then intersexual selection. Intersexual selection is a phenomenon where members of one sex, usually females, choose members of the opposite sex. Between two sexes, males show special traits or behaviors pattern to attract females. Then secondary sexual characteristics. Secondary sexual characteristics, physical features related to the sex of an organism that emerges during puberty or the phase of sexual maturity. Examples of secondary sexual characters is humans include the development of breasts in females and pigmented hair in facial hair in males. Secondary sex characters are distinguished from primary sex characters, which are present at birth and are comprised of the external and internal genitalia, including the penis and testes in males and vagina and ovaries in females. In human females, pubescent changes involve secondary sex characters, including the rounding of the hips and the first phase of breast development. The later begins with an elevation of the areola surrounding the nipple. This slight swelling is caused by the growth of small cone-like structures known as the breast bud. In both sexes, the onset of puberty coincides with the appearance of pubic hair, and this period ends when the pubic hair development is complete. And during puberty, female experience increased breast development with the breast buds enlarged to form the primary breast. In males, the voice deepens and pigmented axillary and facial hair appears usually about 2 years after the emergence of pubic hair. Then changes in primary and secondary sex characteristics are essentially complete in post-pubescence, at which time the individual is fertile. So, nonetheless, some changes in secondary sexual characters continue to occur in this phase. For instance, male may begin to grow a beard, the female may experience further breast development. And the timing of patterns in the development of secondary sex characters may markedly between individuals, often in parallel variations in the onset of puberty. Such variations are influenced by a range of factors, including body mass index, ethnicity, and genetics. The sex hypothesis in evolutionary biology and sexual selection, proposed by Patrick J. Weatherhead and Raleigh J. Robertson of Queen's University in Kingston, Ontario, in 1979, states that a female's ideal mate choice among potential mates is the one whose genes will produce mates with the best chance to reproductive success. This implies that other benefits a father can offer the mother or offspring are less relevant than they may appear, including his capacity as a parent or caregiver, territory, and nuptial gifts. The good genes hypothesis. The good genes hypothesis, or GTH, was formulated by evolutionary biologist W. D. Hamilton and behavioral ecologist M. Zook. It proposes that the characteristics preferred by females are a signal of males' ability to pass on genes, coding that certain characteristics which will increase the survival and reproductive success of the offspring, with the male possessing them. In such cases, sexual selection would lead to the increase of the proportion of genes coding the attractive trait that are beneficial under prevailing conditions. In other words, good genes increase the fitness of the offspring. In an evolutionary and ecological understanding, individuals' fitness depends on how many copies of code